Hello everyone and welcome to the 11th session. In this session we're going to learn more about the neurotransmitters. So we're going to revisit the 4 steps of the synaptic transmission and then see what does it take to be a neurotransmitter, which is a great position. If it's a company, let's consider the brain a company. Neurotransmitters are considered up there. And then what are the different types and structures of neurotransmitters, the synthesis, the classes, and the location. And then I'm going to tell you the specific readings that you will need to focus on. So let's start by the revisiting of the 4 steps. So remember we said that the process of the synaptic transmission involves 4 different phases. The first is the synthesis of the neurotransmitter. And there are different biochemical mechanisms for synthesis, and they differ for each neurotransmitter, as we are going to see in the following slides. And each type of neurotransmitter is packaged and stored in vesicles, which are then positioned in a specific way to be released of their chemical transmitters into the cleft when it's time. The second is the neurotransmitter release, where the release of the neurotransmitter is produced by the action potential when it invades the terminal ending, and then the resulting change in the membrane potential activates the voltage-gated calcium channels, causing an influx of calcium ions into the terminal. And then by mechanisms which are not yet clearly understood again, the calcium ions cause the synaptic vesicles to fuse with the presynaptic membrane, and then the transmitter is released into the synaptic cleft by the process of the exocytosis. And then each synaptic vesicle releases a fixed amount of neurotransmitter molecules. And that's pretty much for the release. And then after that, we have the binding to the postsynaptic neuron, where after the neurotransmitter is released into the cleft, it can bind to a specific receptor that's located on the postsynaptic cell. And last but not least is the termination of the synaptic transmission. And this happens again by either enzymatic degradation or reuptake and recycling of the neurotransmitters into the presynaptic neuron. So these are the 4 steps, and after revisiting those 4 steps, we can conclude that the stimulation and nerve impulses are carried by neurotransmitters. So what are neurotransmitters exactly? They're chemical messengers that are present in the brain, the spinal cord, the nerve junctions. And these substances, as the name indicates, so when you think of it, neurotransmitters, they are meant to transmit or transfer the signals from one neuron to another. As simple as that. So their study is really important in different subjects like anatomy, physiology, pathophysiology of diseases, and also in pharmacology. Why is it important in pharmacology? Because actually there are several drug classes that target either the receptors for the different neurotransmitters, or the synthesis stage of the neurotransmitter, or the termination, and so on and so forth. So different diseases have alterations in different pathways that involve neurotransmitters. That is why when we think pharmacology, we think how can we either generate an agonist or an antagonist of the altered process. So the first thing is, if I wanted to be a neurotransmitter, what does it take? What are the characteristics of neurotransmitters? So neuroscientists have set up a few guidelines or criteria to prove that a chemical is really a neurotransmitter, because not all of the neurotransmitters that you have heard of may have actually met every one of these criteria. So for instance, the chemical must be produced within a neuron. It must be found within a neuronal synapse. It the neuron is stimulated, and when the neuron is stimulated, it must release the chemical. And then when the chemical is released, it must act on a postsynaptic receptor and causes a biological effect. And then after the chemical is released, it must be inactivated, and the inactivation can be either through a reuptake mechanism or an enzyme that stops the action of the chemical. And if the chemical is applied on the postsynaptic membrane, it should have the same effect as when it is released by a neuron. So just to repeat again, there are several characteristics or things that make a chemical a neurotransmitter, and it is as follows. The chemical must be produced inside the neuron. The necessary precursor enzymes must be present in the neuron. There must be enough of the chemical present to actually have an effect on the postsynaptic neuron. The chemical must be released by the presynaptic neuron, and the postsynaptic neuron must contain receptors that the chemical will bind to, and then there must be a reuptake mechanism or enzyme present that stops the action of the chemical. So that is what it takes to be a neurotransmitter. What about the types of the neurotransmitters and their structures? So there are different types of neurotransmitter with diverse functions and roles in the body. They're organic substances which have short span of action, and their action actually lasts for a fraction of seconds at some times. And they can be compared with hormones because they do have similar properties and mode of actions. That is why we can also call them neurohormones. So let's see. The first class or category would be the small molecule neurotransmitter substances. So in this case, we have the acetylcholine, we have the dopamine, norepinephrine, serotonin, histamine, epinephrine. And epinephrine is a hormone actually, which is the adrenaline. And the norepinephrine is also called noradrenaline. And then the dopamine, norepinephrine, and epinephrine are called catecholamines. And this class of neurotransmitters actually are largely targeted by different drugs. So we will see later how the different neurotransmitters are synthesized. But speaking of serotonin, actually it was found that positive self-talk, so when we think positive and talk positive, this releases the serotonin, which is the happy hormone sort of, and the endorphins in our brain, and then flow throughout our body, and that is what makes us feel good and optimistic and so on and so forth. Okay, the second class are the amino acids, like the gamma aminobutyric acid, like glycine, glutamate, aspartate. So these are also neurotransmitters. And in the previous sessions, we discussed, or we knew more about the different receptors, like the glutamate receptor, the GABA receptor, and so on. And there are also the acetylcholine receptors. Then we have the neuroactive peptides like the bradykinin, the beta endorphin, the bombacin, calcitonin, and all other types of neuroactive peptides. And of course, we can see the difference in structure between the different classes of neurotransmitters. And from these slides, we can observe actually that the neuroactive peptides are the ones with the most sophisticated chemical structure. Then we have the soluble gases like nitric oxide and carbon monoxide. So these are basically the different types of neurotransmitters, or let's say the chemical categories of neurotransmitters. What about the synthesis? So in case of catecholamines, like we said, dopamine, norepinephrine, epinephrine, we find that through a series of chemical reactions, the amino acid tyrosine is converted into dopamine, then norepinephrine, or into the hormone, which is epinephrine. And we can find different enzymes in this case. And these enzymes actually could be drug targets as well. What about And this is how serotonin is synthesized as well. Again, through different enzymatic reactions, which means different reactions that are catalyzed by different enzymes. And again, the acetylcholine is synthesized by a reaction between acetyl-CoA and choline, and then we have the choline acetyl transferase giving us acetylcholine at the end. So this is like in brief how the synthesis happens. What about the classes of neurotransmitters? We have the fast excitatory neurotransmitters, we have the fast inhibitory neurotransmitters, and then we have the neuromodulators. So we have excitatory neurotransmitters, we have inhibitory neurotransmitters, and then we have what's called the neuromodulators, whether excitatory or inhibitory. So the fast excitatory neurotransmitters are the most important neurotransmitters. They're fast, and they are like glutamate and acetylcholine, and they communicate strong immediate excitation from the presynaptic to the postsynaptic cell. And they have the most or the longest distance neural projections in the brain, which are made by axons that release glutamate. And all the voluntary muscle contractions are actually caused by the release of acetylcholine by motor neurons onto muscle cells. So they are really important. What about the fast inhibitory neurotransmitters? So because large interconnected systems are made only of excitatory connections, and then they tend to be unstable and subject to seizures, the balance in the nervous system is guaranteed by having inhibitory connections. And that is why the fast inhibitory neurotransmitters are really important. So something like GABA and glycine, which are inhibitory neurotransmitters, they have a very important role in counteracting the excitatory neurotransmission. So inhibitory neurotransmitters allow for computations, and for example, if most of the neurons are voting that you're looking at an elephant, but a couple here and there are saying if it has 4 legs, it could also be a donkey, for instance, then the inhibitory neurotransmitters suppress that it could be a donkey message, because like the more the major amount of neurotransmitters are moving towards that this is an elephant, basically. What about the last class, which is the slow neuromodulators that can include both the excitatory and inhibitory types? So most of the fast neurotransmitters are amino acids, or most of the fast neuromodulators are amino acids. And modulating neurotransmitters also include biogenic amines, such as dopamine, serotonin, and small proteins like somatostatin, substance P, enkephalins, and organic chemicals like the epinephrine and norepinephrine, which are the catecholamines. And then the actions of these transmitters are hormone-like. So they produce slow but long-lasting changes. Yeah, we can think of their action as setting the mood within the brain as a background for what the faster neurotransmitters are doing at shorter timescales. So, for example, we might be resting quietly while digesting a good meal when a lightning storm strikes. And this is considered like a flight or fright condition, right? So this means that norepinephrine, which is a flight or fright neurotransmitter, will be triggered to be released, right? So its action will shut down the digestion and other internal homeostatic processes in favor of increasing our pulse and blood pressure. And this in turn will divert the blood flow to the muscle in preparation for moving quickly to safety. So in this case, the neuromodulation plays an important role. What about the location of the neurotransmitters? So you can have a look at this table and you will find that the different neurotransmitters are located in different regions of the brain. But what we can see here is that glutamate and GABA both are widely distributed in the central nervous system. So of course the slides will be uploaded so you can have a better look at this table with the different locations of the different neurotransmitters. So we're done with this session, and for the readings, please make sure to skim through all chapter 13, but you can focus more on pages 289 to 295. Thank you.